Alright, going to finish chapter 37. Okay, neurons communicate with each other, with other cells at synapses. So remember, those are the spaces between the neurons. At electrical synapses, the electrical current flows from one neuron to another. Most synapses, however, are chemical synapses, in which a chemical neurotransmitter carries information from the presynaptic neuron to the postsynaptic neuron. The presynaptic neuron synthesizes and packages the neurotransmitter in synaptic vesicles located in the synaptic terminal. The arrival of the action potential causes the release of the neurotransmitter. The neurotransmitter diffuses across the synaptic cleft and is received by the postsynaptic cell. Alright, so what does that look like? Here is the presynaptic cell. So this is the cell where the electrical impulse travels down the axon from the axon hillock. And then it reaches the synaptic terminals. Okay, so that's the end of the presynaptic cell. So what happens here is calcium ions will diffuse into the cell, and when the calcium ions diffuse into the cell, what happens is it causes the neurotransmitters to be packaged into vesicles. And then these vesicles will fuse with the presynaptic cell membrane, and it will release these neurotransmitters into the synaptic cleft, or the synapse, the space between the cells. And then what will happen is those neurotransmitters will bind to receptors on the postsynaptic cell or the postsynaptic membrane, and then that will then cause basically ion channels to open on the postsynaptic cell, which will eventually cause an action potential in that postsynaptic cell if the signal is strong enough. Direct synaptic transmission involves the binding of neurotransmitters to ligand-gated ion channels in the postsynaptic cell. Neurotransmitter binding causes ion channels to open, generating a postsynaptic potential. Postsynaptic potentials fall into two categories. Excitatory postsynaptic potentials, or EPSPs, are depolarizations that bring the membrane potential toward threshold. So you're getting possibly closer to threshold, which will cause an action potential to be generated. The other type is inhibitory. So inhibitory postsynaptic potentials, IPSPs, are hyperpolarizations that move the membrane potential farther from threshold. So an example of an inhibitory postsynaptic potential would be, when you go to bed at night, you don't want the neurons in your brain firing. You want to be able to go to sleep. So there are particular chemicals that cause these inhibitory postsynaptic potentials so that you can basically shut down your mind and go to sleep. The duration of postsynaptic potential is limited by rapidly clearing neurotransmitter molecules from the synaptic cleft. Some neurotransmitters are recaptured into presynaptic neurons to be repackaged into synaptic vesicles. Some are recaptured into glia to be used as fuel or recycled to neurons. Others are removed by simple diffusion or hydrolysis of the neurotransmitter. The cell body of one postsynaptic neuron may receive inputs from hundreds or thousands of synaptic terminals. A single EPSP, that's the excitatory one, is usually too small to trigger an action potential in a postsynaptic neuron. Okay, so let's look at these individually. Okay, so looking here, we have an EPSP. We have two of them, right? We have two EPSPs, so we have the terminal branch of the presynaptic neuron, so we have two EPSPs coming in at different times. So because they come in at different times and they are spaced too far apart, then a threshold potential does not get reached. So threshold is not reached, action potential does not get generated. If they come in close together, then what will happen is an action potential will be generated because they have an additive effect. If two EPSPs are produced in rapid succession, an effect called temporal summation occurs. So basically the two EPSPs, their effects get added together, which leads to the generation of an action potential. In spatial summation, EPSPs produce nearly simultaneously by different synapses on the same postsynaptic neuron add together. So they still have an additive effect. The difference is that the EPSPs are coming from different presynaptic neurons. The combination of EPSPs through spatial and temporal summation can trigger an action potential. Okay, so here we have two EPSPs that are acting on one neuron. This is spatial summation. They're acting on the same neuron. They have an additive effect, so an action potential gets generated. You can also have IPSPs, okay? So here's an example of an IPSP and two EPSPs. Now, here we have an excitatory one followed by an inhibitory one. So basically they cancel each other out, okay? And the same thing here. We have an excitatory one and an inhibitory one. So no threshold gets reached because they have opposing effects. Through summation, an IPSP can counter the effect of an EPSP. The summed effect of EPSPs and IPSPs determines whether an axon hillock will reach threshold and generate an action potential. Signaling at a synapse brings about a response that depends on both the neurotransmitter from the presynaptic cell and the receptor on the postsynaptic cell. A single neurotransmitter may have more than a dozen different receptors. Acetylcholine is a common neurotransmitter in both invertebrates and vertebrates. It's vital for functions involving muscle stimulation, memory formation, and learning. The best understood function of the ligand-gated ion channel is in the vertebrate neuromuscular junction. When acetylcholine, released by motor neurons, binds to this receptor, the ion channel opens and an EPSP is generated. This receptor is also found elsewhere in the peripheral nervous system and in the central nervous system. A number of toxins disrupt neurotransmission by acetylcholine. These include the nerve gas sarin and bacterial toxin that causes botulism. Acetylcholine is one of more than 100 known neurotransmitters. So here are some other neurotransmitters. GABA is found at inhibitory synapses in the brain. That's one of the neurotransmitters that allows you to shut off your brain and go to sleep at night. Here we have some other ones. Dopamine and serotonin are two neurotransmitters that are very instrumental in your mood. Norepinephrine is instrumental in your fight or flight response. And then we have some other neurotransmitters here. I wouldn't concern myself with knowing the different neurotransmitters and what their functions are, but you can read these slides if you're interested in learning more about neurotransmitters. So that concludes the nervous system.